Okay, well, we'll get started then. So, hello, everyone. Welcome to the webinar. And I'm Alan Ryan, and I'm a professor of otolaryngology and neurosciences at the University of California in San Diego. I do basic research on otitis media. And what I wanted to do today was to discuss with you the innate immune response in acute otitis media, as it's revealed by experiments that primarily we do, which I'll be talking about. A lot of other people do research in animals too, but I'll mostly be talking about our own data, and as revealed by animal models, and then we will hear about the human otitis media in a little bit. So I'm going to go ahead and share my screen. All right. So let me find the right arrow. Here we go. So as everybody knows, for the majority of children, when you get an initial episode of acute otitis media where you haven't been in contact with that bug before, this resolves within a few days, even without antibiotics. So obviously this is too rapid for cognate immunity to have developed, and adaptive immunity, and implicates innate immunity in the resolution of an initial acute OM. And the cornerstones of innate immunity are pathogen receptors that don't require sensitization to fight infection. Now, this is a very ancient system. It's been around for, many, many millions of years, and it operates across all animals and many plant species. And the foundational family of innate immune receptors are the toll-like receptors, which respond to many pathogen-associated molecular patterns, which are known as PAMPs. Now, in addition, toll-like receptors respond to many damage-associated molecular patterns, or DAMPs. And these are generated when there's damage to host cells, and especially host cell interiors, and broken membranes are flooded across a site of infection. Now, there are many other innate immune receptors that have also been identified, and they provide a wide array of protection from viral and bacterial infections. And this is a diagram of what we see in am I sharing the screen? Are the Zoom pictures showing up and blocking things? Because they are for me.
No, it looks good. Thanks.
Okay, good. So you can see here, the different PAMPs and DAMPs that are responding to the toll-like receptor family. And you can see that there's just a great array of different kinds of molecules that will stimulate the innate immune system. And the activation of these receptors leads to the expression of a lot of effectors that mediate the elimination of pathogens. And those are chemokines that recruit leukocytes, cytokines that are inflammatory or anti-inflammatory. The inflammatory ones are primarily interleukin-1 beta and TNF-alpha, and a lot of antimicrobials, which include the defensins and other molecules. So this is a diagram of some of the systems that operate in innate immunity, with the receptors that are either extracellular, as many of the tolls like receptors, or intracellular, like Pol3, TLR9, and TLR3. There are also other DNA receptors, DAI1 and AIM2, which operate on bacterial DNA. And then there's the NULP receptor, which is a member of the NOD-like receptor family that responds to bacterial proteins. And then there are two NOD-like receptors, NOD1 and 2, which are again intracellular and respond to various pathogen proteins primarily. And those lead to the activation of downstream pathways. There's usually an adapter like ASC or RIP2 or MIghty88 or TRIF that binds to the interior end of the innate immune receptor and initiates different cascades, all of which lead down to different transcription factors that lead to the expression of things like the cytokines, chemokines, and antimicrobials. So to identify the roles of different innate immune receptors and effectors, we have used a mouse model of acute otitis media. And while the middle ear of the mouse is pretty small at 5 microliters, infection with common human pathogens does, produce otitis media that's quite similar to that observed in human patients. And this includes hyperplasia of the middle ear mucosa so that it gets very thickened and recruits different cell types, recruitment of leukocytes, formation of biofilm, and the generation of mucus and antimicrobials. So we study otitis media induced by Haemophilus influenzae, which we abbreviate as NTHI, and we employ knockout mice to explore the contributions of these various immunodomain genes, including receptors and effectors and linking molecules, into the pathogenesis and recovery from OM. All of this is acute OM rather than chronic, so that you should keep that in mind. This includes the deletion of genes that include all of these different possibilities. So this is what, otitis media looks like in the mouse. The normal middle ear is quite simple. It's a monolayer of epithelium with a little bit of stroma underneath. A couple of days after infection, you see the expansion of the mucosa, and in some places becomes a pseudostratified columnar epithelium. It's underlined by a very robust stroma underneath, and there are lots of vessels that have grown in. And then, at 3 days, it's very similar to that. At both 2 and 3 days, you can have a lot of infiltration of leukocytes into the middle ear, both neutrophils and monocytes. Macrophages are the primary cells that come in. But by 7 days, it's gone, and the mucosa has gone back to its more or less its normal state. So these are the knockouts that we've studied to date, which have been circled here in red. So, we've done a number of the toll-like receptors, as you can see. We've done a number of the products, inflammatory products of cytokines and chemokines. We've done some of the adapter molecules, and the NODs families have all been knocked out. So I'm just going to give you an example of otitis media in MIDI88 deficient mice. Now, MIDI88 is absolutely required for signaling from all toll-like receptors except for 3 and 4. 3 uses a related adapter, which is known as TRIF. And while the LPS receptor TLR4 can use either MIDI88 or TRIF. So lack of MIDI88 disables almost all TLR signaling, and we expected its mutation to affect otitis media. And because we know that NTHI can signal through toll-like receptors 1, 2, 4, 6, and 9. So when we look just at mucosal thickness, what you see is that normal thickness drops down by 7 to 10 days. The mucosa has gone to normal. But in MIDI88 mice, it goes out to 28 days. You still have an expanded mucosa. And if you look at the recruitment of neutrophils, you can see in red in a normal mouse that there's recruitment of leukocytes at 1 day, 2 days, and 3 days, and by 5 days they're mostly gone. But with the MIDI88 knockout, you still have very much expanded recruitment that goes out to at least 14 days, and sometimes we even see otitis media at 42 days. So it becomes much more chronic without the contribution of the toll-like receptors. And the same thing with macrophages. Again, 2 days, 3 days, and 5 days, we have macrophages in the middle ear. But, in the toll-like, in the MIDI88 knockout, the macrophage recruitment, which is always later than neutrophil recruitment, extends far out. And again, goes out to 42 days, which isn't shown in this slide. And then we haven't tested beyond that. So we've used the same technique to assess the roles of other genes, including both receptors and effectors. And you can see here, excuse me, I'm going to go back. You can see here what happens with the recruitment with the deletion of different innate immune genes. So in black on the bottom, you can see the wild type mouse, again, recovering by 7 to 10 days. And then, there are all the toll-like receptors here, MIDI88, TRIF, TNF alpha, all produce extended otitis media. And in fact, whenever we have, deleted a gene in the innate immune system, we see extension of the duration of otitis media. And then bacterial clearance is the same. We can see here in the MIDI88 animal that you're still having, positive ears. Out of all the ears that are looked at, 2 out of 6 are still positive at 42 days, and 21 days, 4 out of 6. So we end up with, culture positive bacterial otitis media, which again goes out in a much longer duration, which is consistent with the fact that the phenotype gets longer as well. And this is the results from a number of other things that we've looked at. And you can see that especially TLR2, TNF, and MIT88 have long extension. Some of the other ones, have less so. TLR4 does go out well as well. And the nods, it's a similar kind of picture, not quite as long as we see with some of the tolls. But, again, each one of these has an extension of bacterial culture positivity. So what the knockout experiments reveal, is really the overall role of a gene in otitis media. They tell us which of the various middle ear, they don't tell us which of the various middle ear types contribute to otitis media. So to address this, we've used single-cell RNA-seq to document expression of various innate immune genes by different middle ear cell types throughout an episode of acute otitis media in mice and also before infection. And when we do this experiment, we identify more than 22 different cell types in the normal middle ear. So this is a normal middle ear, mouse. This is a single experiment where we used 6 animals and we took the middle ear mucosa from normal animals. We ran it through the single cell system, and then we ended up with clustering based on gene expression. And you can see there are 17 clusters here. It's difficult to count them, but believe me, there are. But within those clusters, there are some differentiated cell types, especially among the lymphocytes, because there's so many different categories of B and T cells. But, in any event, we see all of these cell types. And here we can see the expression of innate immune genes. You can see the list on the right, which you won't be able to read. So this is just an overall pattern that you're seeing. And you can see that there's one category of cell that really stands out in the expression of innate immune genes. And these are the resident macrophages that live within the middle ear. And they are, primed and ready for the expression of innate immune genes. But if you look at the pattern that you see from, this way of displaying the expression of innate immune genes, pretty much everyone expresses some innate immune genes during the normal middle ear. And that would suggest that the response to bacteria isn't necessarily dominated by the macrophage, although the macrophage must be playing a really important role. So if you can evaluate the expression throughout the episode of otitis media, then you do have a chance to see the role that's played by each cell type in the middle ear immune response to infection. So during otitis media, cell types are added during those observed in the normal middle ear, and the prominent ones are neutrophils and more subtypes of lymphocytes, which are now distinguishable by gene expression patterns. And innate immune responses by cell type are pretty radically different. And this is, another way of looking at the cell type clusters, where 26 cell types and 21 clusters in the infected middle ear. With the, you can see here, the neutrophils in cluster 0 and 4 are major additions to the response. Now, this is the innate immune gene expression of, selected innate immune genes in the normal middle ear. And again, as in the picture, the slide that I showed earlier, the macrophage is, the primary expressor, although every cell type except the melanocyte has at least some expression of innate immune genes. And for example, the epithelial cells on the left, are the best expressors of antimicrobial peptides. Now, that changes a lot when you stimulate the immune system of the animal, the middle ear immune system with NTHI. Now, here's the response that you see, expressed as log to the base 2 of gene expression. And the macrophage and the neutrophil, again, very strong expressors. But you look at all the other cell types except the poor melanocyte, and these are expressing very high numbers of certain of the genes. Obviously, chemokine receptors are dominantly expressed in leukocytes, but the epithelia, the stroma, the endothelium, and the parasites all expressed the receptors, the cytokines, the chemokines, and the epithelium expresses high levels of antimicrobial peptides. So basically, almost all the middle ear cell types make contributions to innate immune defense. Gene expression helps identify the downstream mechanisms by which it produces the phenotype. And that's by looking at the cell types gene expression outside of the innate immune receptor category and looking at things that must be effectors. For example, when we evaluated growth factor genes, we found that HBEGF is a major factor expressed by the epithelium, and that inhibitors will delay or deplete or reduce HBEGF's role in mucosal hyperplasia. So hyperplasia is much reduced in that particular knockout. And here you can see with an adapter, on the left, you can see expression of HBEGF, which is limited to these epithelial cell types. And then on the right, you can see what happens if you, an HBEGF inhibitor. You basically block mucosal hyperplasia caused in this case by poly-IC as a viral analog or with bacteria. So more recently, we've been exploring the regulation of middle ear genes during otitis. We've used a combined method of single-cell RNA-seq and ATAC-seq to identify cell types, and then also regions with ATAC of accessible DNA that are near the genes. And this is a way of looking for, different enhancers, suppressors, and promoter activity in the gene during activity. So this requires not use of single cell, but actual single nuclear RNA and ATAC. So here you can see the cell types that we observe with nuclear RNA, and there are 23 cell clusters, which distribute into the basic categories that we have in the normal middle ear when we use whole cell RNA. Now, this is I'm just going to show you an example that shows regions of exposed DNA that are close to the transcribed regions of 3 genes that we know to be involved in otitis and which are known to increase expression during middle ear infection. The ones that are illustrated are HBEGF, FUT2, and MUC5B, all of which are expressed by middle ear epithelial cells. And we compared ATAC data from the normal middle ear to that observed 24 hours after infection. And 2 of these show changes in accessible DNA within the region of the gene, expressed area, but 1 does not. So here you can see the 3 genes. The way this is displayed, there are, different regions of DNA. Let's take HBEGF. We're looking at 39 kilobases of DNA around the express region, which is right on the bottom of these 3 traces. And that's the expressed region is here, and here are the traces. So these top 2 traces are 24 hours after infection with non-typable Haemophilus influenza, and the bottom trace is an uninfected ear. And you can see that there are 3 areas that are around the gene that are pretty radically changed. So this little area of very mild expression, or mild opening of the DNA rather, becomes much more widely opened and aggressively opened. And this one, which is closed in the normal, is now opened. And when we look at FUT2, again, and below, we have this is the expressed region of the gene. And within the non-transcribed 3' untranslated region, we have 3 areas that are very highly expressed 24 hours after infection, but almost not expressed at all in the normal middle ear. So in this case, I think we're looking at our enhancers that are, suppressors, in this case, suppressors that are decreasing expression in the normal middle ear so that it's allowed to be released with infection. And here we're looking at enhancers probably that increase expression during otitis media. And then in the bottom, we have MUC5B, and at least in this region of the genome, which is, the expressed region is here. Excuse me, I did that by accident. You can see there's basically no change in expression that we see. A little bit higher expression here, but it's not as dramatically increased as some of the other ones. And it's this would be right in the promoter region, which it suggests that if this is a real effect, this effect to this effect, that it should be increasing expression or decreasing expression, which we don't see. So in the mouse, we looked at the question of whether innate immunity might be similar to the human. And what we did was we took normal adult human mucosa, we did single cell RNA-seq, and we correlated the gene expression. And it was very different, very similar to what we see in the mouse. But there were differences, but not for most innate immune genes. And we do plan to perform single cell RNA-seq on pediatric normal and otitis media in middle ears with the COMMeND mentee Jason Quion, who is here at Children's Hospital in San Diego in the near future. So this is the normal human middle ear done by single-cell RNA-seq, 20 cell types, 13 clusters, very similar to what we see in the mouse. And this is a comparison of homology in monocyte macrophages or parasites. And you can see that the correlation coefficients are really quite high. So expression is similar. So in conclusion, innate immunity plays a dominant role in acute otitis media involving novel organisms. Cell structure of the middle ear is quite complex, with becoming a little more so during OM. Virtually all cell types contribute to the innate immune response, and gene expression provides clues to pathogenic mechanisms and potential therapies. And understanding middle ear gene regulation during OM may help us understand the genetics of susceptibility. So thank you very much for your attention, and please join us in Philadelphia for ISOM 2025 in June of next year. I'm going to stop screen sharing. All right. We're not going to have any questions now. I think anyone who has questions or comments on the presentation should put them in the chat, and we will address those at the end of the presentations at the end of the hour. So the next, gives me great pleasure to introduce. Pardon?
No, go ahead. Go ahead.
Okay. It's a great pleasure for me to introduce Dr. Tal Marom, professor of otolaryngology at Ben Gurion University School of Medicine in Israel. And Dr. Marom does groundbreaking basic and clinical research in the role of immunology in otitis, and I'm really looking forward to his presentation on the immune response to acute otitis media clinical perspectives. Go ahead, Tal.
All right. Good evening here from Tel Aviv. Hold on for 1 second. Okay. So can you see the presentation?
We've got it.
You got it. Okay. Hello everybody. Nice to see familiar names here on the on the screen here. So I'm Tal Marom. I'm an otolaryngologist. Okay, and I work for the Samson Asut Ashdod University Hospital, affiliated to the Ben Gurion University here in Israel. And I'll talk about the immune response to acute otitis media from the clinical perspective. So we're not going to talk a lot about genes. I have nothing to disclose, and this is this is the university that I work for, and this is the hospital that I work with. It was opened only 6 years ago. So the focus of this talk would be acute otitis media, which you all know is the most common infectious disease of childhood. It's actually the #1 reason for antibiotic prescriptions in children. It's the 4th reason for pediatrician visits in the US. And when you think about it, we insert more tubes to kids, okay, than we do other surgeries such as appendectomies, hernias, and so forth. So it is a very prevalent disease, and the burden of otitis media is still very high. Now, in order to diagnose otitis media, you need to have a combination of two things. It's the symptoms and the signs. You all know about the symptoms. It's the irritability, the fussiness, the difficulty of sleeping or staying up, tugging or pulling the ear, fever, okay, especially in infants and younger children. Usually I tell my residents, if there's no fever, probably it's not otitis media, it's not acute otitis media. And if there are complications such as tympanic membrane perforation, so you see pus coming out from the ear, and if they have vestibulitis, okay, that's another complication. You might see lots of balance, kids falling, not very steady, okay. And for the long run, okay, but not in the acute phase, children will complain about hearing difficulties and, of course, ear pain. Okay, what about the signs? This is sign in otoscopy, okay. The most predictive sign for acute otitis media is the bulging of the tympanic membrane. What it means is that you see, instead of the normal appearing tympanic membrane, it has like a hill, okay? It's coming towards you. You'll see an opaque erythematous and immobile tympanic membrane if you use the pneumatic otoscopy. And in advanced cases, you might see perforation of the tympanic membrane. And for acute otitis, for acute mastoiditis, you will see the post-auricular swelling and the pinna protrusion like that, okay? And facial nerve palsy, neurological deficiencies, and so forth. But this is quite rare because these are the complicated cases of AOM. So you see here at the bottom a few representing figures. Now, otoscopic findings is this is a very nice picture showing the evolution of acute otitis media in kids. Okay, this is from Paula Tettinen from Finland. Okay, and she actually took pictures. So A is like the normal appearing on your top left, the normal appearing tympanic membrane, and F is the full bulging membrane, and you see the progress. Okay, from just fluids, okay, to start the beginning of the bulging, and then you have full bulging with the congestion of the blood vessels of the eardrum and so forth. But in real life, these are nice pictures to have, okay, but in real life, it is not like that. The management of otitis media is according to your guidelines. I hope you know what I'm talking about. So every country has their own national AOM diagnosis and management guidelines. Okay, when to treat, when not to treat. The bottom line, in mild to moderate cases, you need to watchful wait. You just need to wait. Okay, and probably the acute episode will resolve spontaneously. But in severe bilateral cases, probably you should think about starting immediate antibiotics. Now, nowadays, no specialist is required to make acute otitis media diagnosis. This is unlike previous times, okay, when you had to refer the patient to an otolaryngologist, okay, or maybe even to an otologist, okay, just to diagnosis. And this is really important when we'll talk about the immune responses. So today, no blood workup is needed, okay? It's a clinical diagnosis. I'm here sitting in my clinic, okay? The kid comes with his parents. I do the physical exam, okay? And then I tell the parent it's acute otitis media. I don't take any swabs. I don't puncture the ear. I don't take any culture. And of course, I don't send them for any blood workup. So and because of that, the studies regarding the immune responses for acute otitis media from the clinical perspectives are very limited and not very much. And especially they are focused on the severe cases, the more advanced cases, not on the mild cases where you actually just see the patient, tell them like it will go away like in 1-2 days, or just give them antibiotics and probably you won't see them until the next episode comes and so forth. So no blood workup, no ear taps are required, no nasopharyngeal swabs are taken. And actually, we don't even send, for the acute simple cases, we don't send them for hearing tests, tympanometries, and the clinic, and we'll probably see them only if there's a clinical failure, okay, with after 2-3 days. And these are the cases where we actually think about hospitalizing them and taking a blood test and so forth. So Alan nicely covered the immune system versus the innate versus the adaptive immune systems. So the innate is the non-specific that responds quickly, and as you can see from the timeline here, this one here in the blue, and the adaptive is like the more comes later, okay, in the red, okay, and you have the specific response, and it's usually slowly in the first time where the antigen meets the we have the first encounter. Okay, so what are the innate immune response in for otitis media? So we have the physical barriers. Okay, so the ear canal and the middle ear, they have their own physical barriers like the tympanic membrane, the mucous membranes that help prevent infection. This is why we consider otitis media as a mucosal disease. Okay, it's not a visceral disease; it's a mucosal disease. As Alan nicely showed, it's all about the tympanic membrane. Okay, it's all about the barriers. Okay, it has nothing to do with the bone, the underlying bone. You have the inflammatory response. So upon infection, the innate immune system is activated. Cells such as, and Alan covered that macrophages and neutrophils migrate to the site of infection. They release cytokines and chemokines, which promote inflammation and help recruit additional immune cells to the affected area. And we have also the complement system, which helps to enhance the ability of antibodies and phagocytic cells to clear pathogens. It can directly actually break bacteria and promote inflammation. And we have the adaptive immune response, which is, so we have the antigen-presenting cells, like the dendritic cells, They capture and process the antigen from pathogens in the middle ear. They present these antigens to T cells in the lymph nodes. Okay, this is why sometimes, okay, when you palpate the neck, okay, you see here in the mastoid tip, you probably palpate and feel the lymphadenopathy, okay, level 2 right here. And then you have the T cell activation, okay, I won't get into that, and the B cell response. This is all not very practical for a clinical practice. So how do we have fever? It's a very complex picture. You can see it here, but so we have the immune. We have actually the both arms of the immune system actually participate. And I won't get into all the details and the little details here, but what I'm showing here is that in order to have a fever, you need to have a very strong inflammatory response within the middle ear cavity. The middle ear cavity and the mastoid cavity, the bone right here, okay, are connected via a tiny corridor named the Otitis ad antrum. So when we look at the studies that actually looked at the immune response as we measure it in blood test, okay, there are very, very few. And I'll actually show you some of our studies that were performed almost 10 years ago. And we show that the pneumococcal acute otitis media, for example, is associated with higher white blood count levels and higher C-reactive protein levels than a non-pneumococcal AOM. And why is it important? Because in recent years, we've seen the introduction and implementation of pneumococcal conjugate vaccines, okay, into national immunization programs. So what you can see here, if you have a pneumococcal disease, okay, there is a higher white blood, higher WBC levels, okay, in both boys and girls. What about absolute neutrophil counts? Does it help? Some say they do, as you can see here. So these are the blood test, the results of blood tests obtained from kids that had complicated mastoiditis, which is the more advanced acute otitis media, and they show that the higher your absolute neutrophil count is, the higher is your likelihood to complicate with acute mastoiditis, and of course undergo surgical procedures such as mastoidectomy, drainage of abscess, and ventilating tube insertion. What about CRP levels? Again, these are numbers from our previous works. Again, if you have a pneumococcal disease, your likelihood to have a higher CRP level is higher. I'm sorry, you will probably have higher CRP levels in both boys and girls. And actually, this was one of the questions that I sent you. So what is like the most predictive blood test that can help us in predicting the severity of acute otitis media? So actually, this paper that was published recently in the Frontiers of Pediatrics actually meta-analyzed many studies of infections in children and showed that if you have a CRP level of more than 20, okay, this can be considered as a cutoff for starting antibiotics for otitis media and for other respiratory infections. So if so CRPs actually are, we will probably use that, okay, as a cutoff, okay, when we consider starting immediate or antibiotics or not. So think about it. What about the multi-drug resistant strep pneumo? So we are in the era of PCVs long time ago, and what we see now is serotype shift. We are now at the era of PCV13 and PCV10, and if you know, our next vaccines are right there, okay, next door, PCV15 and PCV20. Maybe we'll talk about it in Philadelphia. And the more vaccines that we introduce, okay, the more pressure we give on the bacteria, and they tend to be much more resistant to antibiotics. So if you have a multi-drug resistant strep pneumo, okay, probably you will have a more severe inflammatory response, as you can see here from your blood workup, okay, and you probably have a higher incidence of mastoiditis. So this is very interesting, okay. if you were able to identify, to isolate strep pneumo in the ear, okay, and you have these values, okay, you should be very careful, okay, and very cautious with those kids. What about new systemic markers? Okay, so they're out there, and if you read the literature, you'll probably see markers such as the NLR, the neutrophil to lymphocyte ratio, and probably you'll see the PL out there, platelet to lymphocyte ratio, and you'll even see the SII, the systemic immune inflammation index, okay? And all of them have been shown to have very good prognostic roles in severe infections other than otitis media, such as pneumonia and cellulitis. Okay, but do they have the role in otitis media? And actually, I'm giving our mentees here an idea to conduct a study. The bottom line is no, we don't have sufficient data. So what is the neutrophil to lymphocyte ratio, the NLR? Okay, you just divide the number of neutrophil into numbers of lymphocytes. So 1 to 2 is your normal range, and the higher your ratio is, the worse you have a worse outcome. Okay, this has been shown in many infections, even in hepatitis and so forth, and as I said, in pneumonia. So what about the otitis media? I just found one single study, okay, coming from Turkey. Remember these names, okay? This is a group from Turkey, and they actually published a few studies, okay, on the topic, okay? And they show that you can actually tell or predict, okay, the nature of the fluid in the middle ear. Would it be serous or mucoid, okay? If you actually look at the NLR. So if you have a higher NLR, you probably tend to have more serous fluid in the middle ear. If you have a lower, then you probably have a mucoid. And for controls, you see that the values it all came out statistically significant. So the NLR has been shown to predict the viscosity of the fluid within the middle ear. What about the platelet albumin bilirubin index? Okay, the PALB. I didn't mention it in my previous slide. The bottom line, it's probably only useful in liver diseases. Okay, not in our otitis media. Okay, but in complicated cases, and actually we're trying to extract the data now from our own de-identified huge databases that we have here in Israel. It might be also a useful indicator to predict the severity of disease. So once the kid is being admitted to the hospital, you probably take chemistry, okay, and blood count and so forth, and you can actually predict, will this kid, okay, deteriorate and have mastoiditis and need surgery, okay, right here, right from day 1, from day 0, actually. So as for now, I looked at the literature last week, so there are no studies for PALB. What about the PLR? Okay, the platelet to lymphocyte ratio. This is usually valuable for cardiovascular diseases, okay, because you have here the platelets, and it actually can predict whether you'll have stenosis, even an emboli, okay, even a heart attack, okay. The same group from Turkey also showed that the PLR is also valuable, okay, for predicting the viscosity of the fluid within the middle ear. I have to tell you, they are the only group that actually were able to demonstrate those measurements and results. So this is an idea for you. What about the SII? Okay, the systemic immune inflammation index. Okay, again, the same group from Turkey. One study, they showed also this marker can also be valuable. So think about it, about these new markers that actually can help us predicting the severity and even the course of otitis media. Once the kids are being hospitalized, again, these are for the more advanced cases. Okay, what about eosinophilic otitis media? I listened to Alan's talk, and he didn't mention, right, Alan? He didn't mention about the role of eosinophils.
No, I did not.
Okay, So this is an entity that I have to tell you that I never diagnosed it, okay? I've never seen even one kid about it. But when you look about the mounting numbers of reports about eosinophilic otitis media, it's probably more common in the literature than in real life. So what people say? So what do people say about it? So these you have here the H&E slides, okay, and they show you that the abundance of eosinophils within the middle ear mucosa and so forth. Actually, so it is connected to those hypereosinophilic syndromes such as Churg-Strauss and Leffler and so forth. And eosinophilic otitis media actually often present with symptoms similar to other types of otitis media. And this is why it's very hard to differentiate AOM and EOM. Okay, and you have, but however, it may also present with more atypical symptoms or with a persistent course despite treatment. The diagnosis is very challenging. It involves a combination of clinical evaluation, otoscopic examination, and sometimes more invasive procedures like tympanocentesis. But I told you that usually we don't perform those procedures in simple cases, so probably we'll never know. How will you know? Probably if you give antibiotics and the otitis is still there, okay? Then you have to think about maybe it's eosinophilic otitis media. So probably the diagnosis is per exclusion. Sometimes you have to use corticosteroids or other immunosuppressive therapies to reduce eosinophil activity and inflammation. And allergies may be also managed if they are contributing to the condition. Again, I have to tell you, and I have a very busy pediatric clinic. I've never seen even one case of eosinophilic otitis media. The prognosis varies, so sometimes it's a yes and sometimes it's a no. The last part of my talk would be about the role of breastfeeding and otitis media protection. Okay, I'm sure you all know about the protective values of breastfeeding. There is a debate for how long. Should it be an exclusive breastfeeding, or can we combine it with formulas and so forth? The bottom line, of course, it's one of the most protecting, not a procedure, one of the most protective things that you can actually avoid otitis media in your infant, okay? And how's that? Because of the passive, okay, transfer of immunoglobulin from the human milk, okay, to the baby. Not only that, okay, so here you have a list of all the good things that you have in the mother's milk, okay, and it is important to remember that the composition of the human milk changes throughout time. So you have the colostrum, which is the milk for the very first few days. Then it changes to the transitional human breast milk. And then you have the mature term, and you see the composition is different, okay? You have different cytokines, the different cells, and different immunoglobulins. This is another question that I ask you, but what is the most important protective agent that the mother transfers to the baby? It's probably microRNA, okay? I won't get into it because of the limit of time, but new evidence have shown that microRNAs, okay, passively passing from the mother to the baby are actually the most protective material that actually helps to prevent acute otitis media. There's a lot to talk about. Maybe we can do it in ISOM. And thank you. I've been to Philadelphia. It's a great city. So come and see us all, and let's meet in Philadelphia. And do feel free to contact me. You have here my email addresses and so forth. Thank you.
So thank you very much, Tal, for a wonderful presentation. And so we need to move on to our next speaker, Dr. Nikki Johnston, who's professor of otolaryngology, microbiology, and immunology at the Medical College of Wisconsin. Now, Dr. Johnston will be presenting the most important topic of the webinar, and that's how to write successful academic and industry grants. So, Nikki, take it away.
Can you see my slides?
Yep, we've got them.
Yeah. So hi, everyone. First of all, I'd like to congratulate Dr. Santos Cortez and the team on the COMMeND program. It's really an honor to participate and join you all today. So I am from Scotland originally. I've been in the US for about 20 years now. Originally worked at Wake Forest University in Winston-Salem, North Carolina. And I've been at the Medical College of Wisconsin in Milwaukee, Wisconsin for about 17 years now. I'm a professor in the Department of Otolaryngology and Communication Sciences, and I have a secondary appointment in the Department of Microbiology and Immunology, which is where my research lab is located. So my team are leading a drug development program to develop a new therapeutic for patients with reflux disease that specifically targets pepsin, so a little bit different from what most of you do. But our team also have the pleasure of working with our dean, Dr. Joe Kirshner, on his otitis media research program. So I'll share a little bit about that and where we've been able to help in that field. So Reggie asked me to spend the first 15 minutes telling you a little bit about my research, and then to share like an overview of grant writing, which I know is everyone's favorite topic. So first of all, I'd like to disclose that I'm co-founder and chief scientific officer of Enzyme Biomedical, and venture capital VC investment in Enzyme is funding drug reformulations of fosamprenavir and clinical trials to assess its efficacy for the treatment of reflux disease. So I'm sure most of you are aware, gastroesophageal reflux is the backflow of gastric contents up into the esophagus, which causes heartburn and esophagitis. Gastroesophageal reflux disease, GERD, is the primary risk factor for Barrett's esophagus, which is a pre-malignant condition which carries an increased risk of developing esophageal adenocarcinoma. More proximal reflux up into the throat, up into laryngopharynx, is called laryngopharyngeal reflux, or LPR. And symptoms of LPR include chronic cough, chronic throat clearing, globus, which is a sensation of lump in the throat, dysphagia, which is problems swallowing. But chronic LPR can also contribute to more severe life-threatening illness, such as reactive airway stenosis, and laryngeal cancer. So the reported prevalence of GERD and LPR in the United States is over 67 million and 83 million respectively, with approximately 40% of these patients experiencing overlapping symptoms and disease. Diagnosis and treatment of reflux has really been focused on acid for the last couple of decades. However, development of new technology called combined multi-channel interluminal impedance has revealed that the majority of reflux episodes that get up into the laryngopharynx are in fact not actually acidic, but these non and weak acid reflux events are associated with symptoms in acid-suppressed patients and are resolved following anti-reflux surgery. So therefore, one of the non-acid components must be responsible for those persistent symptoms and endoscopic findings. And so the vast majority of patients with LPR do not respond to potent acid suppression PPI therapy, but also 40% of GERD patients still have refractory symptoms on these medications. Pepsin, as I'm sure you know, is the main digestive enzyme in the stomach. It's present in all gastric refluxate, frequently detected in tissue and secretion samples taken from patients with GERD and LPR, but absent in impedance-confirmed reflux-free control subjects. Non-acid reflux of pepsin is associated with symptoms and endoscopic findings, and physiologically relevant concentrations of pepsin cause inflammation and damage in vitro and in vivo studies and result in pathological changes that are consistent with what is seen in LPR patients. So given this and the inefficacy of PPIs for the treatment of LPR, and it's a very prevalent disease, a new treatment which specifically targets pepsin could be of great value. We found that select HIV protease inhibitors bind to the active site of pepsin and inhibit the enzyme. So the high throughput screening assays that we did, as well as the binding and activity assays, were all funded by a seed grant by the Medical College of Wisconsin's Therapeutic Accelerator Program. So this is an example of how relatively small seed grants can give valuable data to support larger funding opportunities. So we showed that fossamprevir, one of the HIV inhibitors, binds to and inhibits pepsin and prevents pepsin-mediated inflammation and damage in an in vivo mouse model of LPR. In support, patients taking these HIV inhibitors have a significantly lower incidence of LPR compared to the general population. Using MCW's clinical data warehouse, we showed that only 0.2%, 5 out of 2,062 patients taking HIV inhibitors had any record of reflux in their medical record, compared to an age, sex matched cohort, or that of the general population, which is reported at 10 to 34%. So while limited, this epidemiological data does support our hypothesis. Fossamprevir, as an HIV inhibitor, targets a foreign virus, so it's not going to have unwanted additional side effects. It has a really good safety and tolerability profile, so it's an ideal drug to repurpose and to reformulate, allowing a safe assessment in a clinical trial. And this repurposing approach allows for a more expeditious assessment in a clinical trial, of course, compared to that of a new molecule. We've created a new sustained release formulation of oral fossamprevir. Our formulation contains a sodium alginate, and this basically increases mucoadhesion, prolonging drug delivery to the esophagus when swallowed. And so we hypothesize that this will have improved efficacy for the treatment of reflux. This schematic shows an overview of our 12-week randomized, double-blinded, placebo-controlled trial to assess the efficacy of oral fossamprevir for the treatment of LPR. I co-founded Enzyme Biomedical with my business partner, Franco Vigili, who's in Canada. An investment in Enzyme is, as I mentioned, is funding the clinical trials to assess the efficacy of our formulations of fossamprevir as a pepsin inhibitor for the treatment of reflux disease. We're currently finalizing a commercial investigational new drug application with the FDA. You thought the NIH was bad. This is a whole new level of pain. And so we have actually hundreds of people signed up to participate in the trial. So we're excited to hopefully start that within the next few months. We got funding from the Falk Medical Trust, which I'll tell you a little bit more about later, but a Catalyst Award funded drug reformulation for local treatment by dry powder inhaler. So of course the larynx and the surrounding tissues affected by LPR are readily accessible, allowing a higher concentration of drug delivered at the disease site and lower systemic concentration. So this will likely increase efficacy and decrease systemic side effects. So we showed that aerosolized, that local fossamprevir, prevented all pepsin-mediated inflammation and damage in an in vivo mouse model of LPR with an inhaled mass of only 0.93 mg per kilogram per day compared to 20 mg per kilogram per day of oral fossamprevir, again supporting the potential efficacy of a low dose of fossamprevir delivered direct to the throat. Aerosolized local fossamprevir appears not only to be highly effective but safe. Obviously, this toxicity of oral fossamprevir, given that it's a HIV inhibitor, been used for many years for the treatment of that disease, is already known. But of course, this drug has never been inhaled before. So we did pre-GLP inhalation toxicology studies, and we showed that there's no pathology in the nose, larynx, esophagus, etc. No inflammatory cytokines or cardiac toxicity markers were elevated in bronchial lavage or plasma samples. So again, holding promise for the local treatment of LPR with a low dose of fossamprevir delivered by dry powder inhaler. And then as far as we're aware, all dry powder inhalers currently on the market target the drug delivery to the lower airway. So we've been working using computational fluid dynamic modeling to look at the particle size to kind of maximize an inhalation rate to maximize drug delivery to the laryngopharynx. And so we found that a particle size of 9 to 12 microns aerodynamic diameter is optimal for higher deposition in the larynx and lower deposition elsewhere in the respiratory tract, with an inhalation rate of 30 to 60 liters per minute. So GLP inhalation toxicology studies of fosamaprevir funded by a Falk Medical Trust Transformational Award are currently being funded by and performed by Crea Medica. And this will enable an IND application for a phase 1 clinical trial of a fosamaprevir dry powder inhaler. So we're excited to start a phase 2 proof of concept clinical trial to assess the efficacy of oral fosamaprevir within the next few months. And then, all going well with our GLP inhalation toxicology studies, we'll perform a phase 1 clinical trial of a fosamaprevir dry powder inhaler. So I encourage you to explore the Falk Medical Trust funding opportunities. Their Catalyst Awards provide funding at $300,000 for a year, and then successful applicants, awardees are eligible to apply for a $1 million transformational award, which can either be over 1 or 2 years. And both of these are for preclinical studies. So how did I get from there to otitis media research? So before our drug development program kicked off, our team collaborated with multiple physicians and surgeons to measure pepsin as a biomarker for reflux and aspiration in clinical samples. And so one of those studies was with Dr. Joe Kirshner, I'm sure you know from the otitis media field. And so similar to Tasker et al., we found high levels of pepsin in middle ear effusions taken from kids with otitis media. And then we hypothesized that obviously the pepsin up in the middle ear, but given the pH, would likely be inactive, but it is stable below pH 8. So we hypothesized that inactive but stable pepsin and bacteria in the middle ear could have a synergistic effect. So as you know, two of the most common bacteria involved in OM, strep pneumoniae H flu, both produce acidic metabolic byproducts, so potentially activating any pepsin present. We then went on to publish several studies where we looked at an association between pepsin and the inflammatory signaling and effusion viscosity in pediatric otitis media. Interestingly, reflux of pepsin is not only common in otitis media, but it is associated with poor outcomes. It's associated with the need for multiple tube placements. So Dr. Kirshner and I then went on to get an R21 from NIDCD to using our team's experience with cell culture to establish human middle ear cell lines as tools for the otitis media research community. And so our work suggested that pediatric cell cultures generally exhibited an enhanced inflammatory response to the OM-associated stimuli relative to the adult-derived human middle ear cell cultures that were available that were established from an adult cochlear implant patient. And so these may be an improvement upon the current human middle cell culture model. We subsequently immortalized these primary cell cultures, which are described in Mana's paper, which is on the bottom left-hand corner of this slide, then immortalized them with a temperature-sensitive SV40 T antigen and licensed them to ABM and ATCC. So if our team can provide these cell lines to you or help with any of sort of this cell culture type research in OM, just let me know. We're happy to do so. So I know it's a little bit of a change between sort of otitis media and reflux, but so in this part of the presentation, I'll review some of the different types of grants and funding agencies, go through some of the key elements of a grant proposal, highlight some common mistakes, and share some tips to hopefully help you succeed. I am no expert on grant writing. It is my least favorite part of being a scientist and definitely one of my personal weaknesses, but it is a necessary evil that we all must learn to be successful. So there are several types of grants available, largely broken down into three categories: federal grants, private foundation grants, industry grants. Federal grants such as the National Institute of Health, NIH, or the National Science Foundation, NSF. NIH is the largest funding agency in the US with a budget with over, I believe, 47 billion this year. NSF provides funding for research in science, technology, engineering and math, so in the STEM fields, and NSF's budget is, I believe, 11 billion for this fiscal year. The Gates Foundation provides funding for medical research, global health initiatives and education programs. It's a budget of over 8 billion. There are many private foundations, so of course one of which I mentioned earlier, which supported our drug development program is the Falk Medical Trust. And as I mentioned, their Falk Medical Trust Catalyst Award provides up to $300,000 for a year, and then successful applicants can apply for a subsequent $1 million transformational award. And that's been a really great foundation to work with. Industry grants, of course. I know that at least initially in my career, there was this, academics on one side and industry on the other. I think thankfully they're being a bit more collaborative going forward. But I highly encourage you to explore potential industry partners with shared interests and goals. Our team have had a very positive experience working with companies such as Reckitt's on mutually beneficial collaborative translational research projects. Seeing their different perspective from industry is been very valuable. It's often a quicker turnaround time, and you can establish a long-term relationship with a company to move a project from bench to bedside. Networking with other researchers in your field, which I think is one of the reasons the COMMeND program is so valuable, can really provide valuable insights into the funding landscape to help identify funding opportunities, of course, as well as learning a bit more about the latest research in this field. Seed grants, typically at a smaller funding level, provide funding for pilot research projects. Applying for seed grants can provide valuable experience in grant writing and project management, contributing to professional development, which can help you prepare for future larger grant applications and studies. My seed grant from MCW's Therapeutic Accelerator Programme was an invaluable resource, and not only just for the funds for the preclinical studies, but provided access to a network of experts in drug development. So I encourage you to utilise your local institutional resources to support preliminary studies, after which you'll hopefully be more competitive for national larger funding opportunities. Here's some funding sources which have made specific announcements for research in otitis media. By no way an exhaustive list, just a few options for you to consider. I'm sure these slides will be shared. And in this section, I was going to discuss just some of the key elements of a grant proposal. You also need to prepare an abstract, which is a brief summary of the proposed research. It should clearly and succinctly describe the research question, hypothesis, methodology, and outcomes. The specific aims section is really the backbone of a research proposal. It outlines the research question and goals, provides a clear and concise overview of what the research aims to achieve and how it will contribute to advancing knowledge in the field. I think the specific aims section is the most vital part of any grant application. In this section, you must quickly gain the reviewer's trust and confidence while simultaneously convincing them that your work is important to fund. You've also got to convey that you and your team are the best people to complete the work that you proposed. So for these reasons, it can be one of the most difficult sections to write. It's really central to your grant proposal, therefore should probably be the first section that you write. You can think of your specific aims page as an abbreviated version of your full grant application. So by having this specific aims page written well, it will help the rest of your grant application. I think will be easier to write. I've included a link at the bottom of this slide to an article which I think really provides some great tips on structure, content, and organization of a specific aims page. The background and significance section provides a rationale for the proposed research. It should outline the current state of knowledge in the field, the gap in knowledge that needs to be addressed, and why the research proposal is important, how your results will contribute to the field. Address the strengths and weaknesses of prior research used to support the application, and describe how your proposed research will address weaknesses and gaps in the field. The innovation section highlights the potential impact of the proposed research and how it will advance the field. It should highlight your innovative ideas. What's innovative about your study? Is it the novel hypothesis? Is it the approach, the technique, treatment, collaborative team working to address the problem, etc.? In this section, demonstrate how your proposed research is novel, how it explores new scientific avenues, has it a novel hypothesis, explain how your application or to support a new application of an existing concept or method. If your research is paradigm shifting, if it challenges commonly held beliefs, be sure to include sufficient evidence in your preliminary data to convince the reviewers, including a strong rationale and data supporting your approach. You need to make the reviewers feel confident that the risk is worth taking. Preliminary studies demonstrate competency in the methods and interpretation. Well-designed and robust preliminary studies also serve to provide a strong scientific rationale for the proposed follow-up experiments. Reviewers will use the preliminary studies together with the bias sketches to assess the competence of the research team. The research strategy section provides a detailed description of the research project, including the study design, population, data collection and analysis methods. This section outlines the procedures and the techniques that we use to address the research question. Don't forget to lay out a plan for alternative experiments and approaches in case you get uninterpretable or surprising results, and also consider limitations of your study. Expect the reviewers to scrutinize your approach. They'll want to know what you plan to do, how you plan to do it, and whether you can do it. NIH data shows that of all the peer review criteria, the approach section has the highest correlation with overall impact score. Grant writing, of course, involves a degree of ethical responsibility too. It's important to ensure that the research is conducted in an ethical manner, and so disclose all relevant conflicts of interest. Grant proposals should be clear, concise, and well organized. The language should be accessible to a broad audience, and technical terms should be well defined. The proposal should be easy to navigate with clear labeled sections and subheadings. An effective writing style is essential for a grant proposal. Clear and concise presentation can make it easier for grant reviewers to understand your research and therefore see its potential. Grants should be formatted according to the funder's guidelines. Review the instructions multiple times. It's important. Use font sizes, margins, spacing, read the instructions. Formatting can impact the chances of a grant getting funded. It ensures that the proposal is easy to read, visibly appealing, and that it meets the funder's guidelines. You don't want to annoy your reviewers. Grant proposals should be reviewed and revised multiple times before submission, so allow plenty of time for those revisions. This can be done by colleagues and mentors and grant writing consultants. Perhaps your COMMeND mentor would be willing to review your grant application and provide feedback for improvement. Common mistakes in grant writing include not following the guidelines, incorrect budgeting, lacking significance. The approach doesn't include a testable hypothesis, weak or absent preliminary data, overly ambitious aims, aims that hinge on the success of a previous aim, untested or overly complex methodology, absent statistical justification, lacks innovation, or investigators lack experience or collaboration. And finally, some tips to help you succeed. Be concise, keep your writing brief, clear, and understandable. Be accurate. Include realistic plans to address problems. Only commit to activities that you can complete. Tailor your proposal. Each grant opportunity is unique, so treat it as such. Write to persuade, convey a compelling core idea and communicate your message clearly. Use visuals where possible. Include visuals to help you illustrate your points. Leave white space. Don't clutter your pages with small margins, small font, or too many words, like my slides. Get it proofread. Have someone who isn't familiar with your research read your proposal for clarity. Meet deadlines. Start early to allow time for review and revisions before the proposal is due. And then one of the important lessons that I learned early is that of fit. I submitted several applications to NIH for our drug development program, and just realizing it wasn't a good fit. So all refluxulated applications tend to get targeted to NIDDK, but our target patient population is actually more relevant to NIDCD. So it fell between the cracks, so to speak. So finding funding for the preclinical work, both internally using MCW's Therapeutic Accelerator Program and then the Falk Medical Trust, allowed us to get this drug development program to a point where we could start a new company, and then the VC investment in that company will fund the clinical trials. So I think the audience is as important as the proposal. And where there's a will, there's a way. Funding requires persistence and patience. With that, I'll stop, and happy to take any questions.
All right. Now I know that in the chat where there was supposed to be, some curation of any of the questions and so forth. So we can look at the chat. If I can find it, here we go. Okay, let's see. Okay, Andrew Cox asks a question to everyone. Does the co-occurrence of viral infection in virtually all pediatric AOM explain the innate immune cell differences that you see between mice and humans? I think that probably can best be addressed by me. And, I think that there's no question that when you have co-infection, that it produces a different set of immune mediators than when you have bacterial infection. And we have done that experiment by putting in, influenza A as well as NTHI into the middle ear of mice. And you do see, quite dramatic enhancement of the infection. It has to be noted that, any study like we do in animals, no matter whether it's mice or chinchillas or guinea pigs, these are not normal commensals of these animals. So they are going to have, a radically different response than humans in some ways, because humans are adapted to these, and the bacteria are adapted to the animal, to a human. That said, we do see quite a lot of similarity. And as Tal points out, it's hard to do experiments on acute otitis media in humans. So, the use of the animal model has utility there, but it should also be taken with a grain of salt that, things are different. But, when we look at our models, and we look at, the difference between humans and mice, there's much more similarity than there are differences. So I hope that answers the question. Okay, let's see what else we have. Okay, that was the only question in the chat. So I think everything was so clear, except for mine, of course, that there weren't any questions that came up. So I do want to thank everybody for attending the webinar, and I hope that you've enjoyed this experience. And I also wanted to thank Nikki and Tal for wonderful presentations. And, we really appreciate the time and effort that you put in to come and educate everyone who's attending here. So Reggie, do we have any other issues that you would like to have covered?
Thank you for all the wonderful talks. They were really excellent. It just I hope that's really helpful to all our COMMeND mentees, especially the grantsmanship and all the technical content. It also sets us up well for the next webinar. Please watch out for announcements for that next webinar, which will tackle medical treatment for acute otitis media and also lab management. Thank you everyone for joining us today. We will also have announcements when this webinar is posted on the website, so that if you want to review any of the information, then you can have access to that.
All right. Well, thank you very much. And Nikki, it sounds to be like you would be willing to review my next grant application. So
I think I'm more sending mine to you.
Thanks, everybody. Goodbye.
okay, take care, everyone. See you at the next webinar.
Have a good day.
Bye-bye.